This method permits to detect, by a non-invasive approach, patients with colorectal neoplastic lesions. In particular, this test permits, in association with tests currently used in colorectal cancer screening program, to better predict the presence of cancer or high-risk adenoma lesions.
To begin this protocol, centrifuge an aliquot of each positive control, standard DNAs, and spike-in DNA in a dry format for 10 seconds on a mini-centrifuge. Resuspend the positive control with 750 microliters of water. Resuspend the spike-in DNA, used as an exogenous internal control to test the presence of inhibitors, with 100 microliters of water. For the standard curve, resuspend the dry standard with 100 microliters of water and make 4 1:5 dilutions starting from the stock solution. Then carefully vortex the reagents for 15 seconds and centrifuge in a mini-centrifuge for 10 seconds. For complete resuspension, store the liquid reagents at room temperature for 30 minutes before using. To prepare the 1x spike-in DNA control, mix 5 microliters of FLDNA spike with 45 microliters of sterile water. For the samples, mix 75 microliters of each sample with 50 microliters of 1x spike-in DNA, yielding a total volume of 125 microliters. First, open the qPCR operating software.